Welcome to this guide on gel electrophoresis, a fundamental technique used in molecular biology for separating macromolecules. Gel electrophoresis separates molecules like DNA, RNA, and proteins based on size and electrical charge. Nucleic acids are negatively charged and migrate towards the positive electrode through a porous gel matrix. Before starting the procedure, let's quickly review the necessary materials and crucial safety points. You'll need an electrophoresis chamber, power supply, agarose, buffer, a DNA ladder, samples, loading dye, and visualization equipment. Remember to handle ethidium bromide, a common stain, with extreme caution. Also, always follow safety guidelines when using the UV transilluminator for visualization. The first major step is preparing the agarose gel, which acts as the separation matrix. Determine the required agarose concentration, weigh the powder, add electrophoresis buffer, and heat until the solution is completely clear and dissolved. Allow the solution to cool to around 50 to 60 degrees Celsius. If using an integrated stain, such as SYBR Safe, gently swirl it into the cooled liquid gel. Next, we cast the liquid agarose into the tray to form the gel with wells for sample loading. Set up your casting tray by sealing the ends and placing the comb in position, typically near the negative electrode end. Carefully pour the cooled agarose solution into the tray, avoiding bubbles. Let it solidify at room temperature for 20 to 40 minutes until opaque. Once solid, gently remove the comb to create the sample wells and remove the casting tray seals. Now, we place the gel into the chamber and run the electrical current to separate the DNA fragments. Place the gel in the chamber with the wells near the negative electrode. Pour enough electrophoresis buffer to completely submerge the gel, about 1 to 2 millimeters above the surface. Mix your DNA samples with loading dye to help them sink into the wells. Load the DNA ladder first, followed by your prepared samples into the remaining wells. Secure the lid, connect the leads correctly, black to black, red to red, and set the voltage, typically between 80 to 150 volts. Turn on the power. Monitor the tracking dye's migration. Stop the run when the dye has traveled about 75 to 80% of the gel length. The final stage involves visualizing the separated DNA bands and interpreting the results. If post-staining, immerse the gel in the staining solution, then place the gel on a UV transilluminator to visualize the fluorescent DNA bands. Document your results by photographing the gel. Analyze your sample bands by comparing their migration distance to the known fragment sizes in the DNA ladder. Gel electrophoresis is an essential technique for separating macromolecules by size. Master this procedure to advance your molecular biology research.